I'm going to be talking about opportunistic data structures with applications by Ferragina and Manzini. So I work as a software engineer for a bioinformatics company. So I'm just going to give a bit of background as to what bioinformatics is. At the heart of bioinformatics is DNA. It's this really long molecule made up of 4 chemicals, and we use the symbols A, T, C, and G to represent them. We have machines called DNA sequences which read the DNA and digitize that string. And all this digitized data needs to be processed, analyzed, and stored. And as you can see, it's really a multidisciplinary field. There's chemistry involved, there's engineering in building the machines, and there's obviously computer science and software engineering required to deal with all that data. Now, unfortunately, DNA sequences, they're not that nice to play with. You know, they give you very short reads rather than the entire string. So depending on the machine, it might be a 30 symbol long read, or it could be up to 1,000. And those reads can also have errors or multiple errors in them. And software has to be able to handle or correct for these shortcomings of the machine. So just the typical workflow, you'd have DNA in a test tube or somewhere. You put it into a machine, and out comes some short reads. So in this example here, the reads are only 4 symbols long, and that's not very useful for us. It doesn't look anything like the original DNA. We'd have to do some processing. And usually that's either assembling those reads into the original long string, or alignment, which would mean taking those reads and a reference and trying to place them on the reference to figure out what it is. So we're going to talk about alignment, and that's what brought me to this paper via another paper. We want to classify an organism from its reads. We might have a big database of, say, all the nasty disease-causing bacteria, and we take our reads and we try and match them against all of those genomes. There are some really good tools out there to do that. One of them is called Bowtie, and Bowtie relies on a thing called the FM index, which is what's described in this opportunistic data structure paper. So DNA alignment is a little bit like a game of Tetris. You take a read and a reference, and you try and find where that read belongs. So in this case, we can slide the read along and we find, oh, looks like a match is there. So we drop it in place. We take our next read. And this one here, it doesn't seem to match anywhere. That's an interesting result. We've got something that doesn't belong to this genome. We take another read. And again, we find somewhere where it belongs. And we slowly build up this picture of reads mapped to the genome. And not everything might be covered. You can see here there's 2 regions that don't have anything covered. So we might say, well, the thing we sequence looks a little bit like this bacteria or this turtle, but it's not quite it. So we might try doing this process again with a different reference. The human genome, for example, is 3 billion bases. It's around 750 megabytes. The sequencer produces millions of reads. In that Tetris example, we sort of stack the reads up to maybe 5 times. But bioinformaticians tend to like their reads, say, 20 high, because there are so many errors and they want to try and correct for that. So each mapping step in this example takes n time, where n is the length of your read and n is the length of your entire genome. And as you can imagine, this gets even worse if, say, you've got, I don't know, 1,000 humans in the database. It's not great. So one solution might be to use an index. And the index we're going to use is the suffix array, because it leads nicely into this paper. So with the suffix array, we take a string, such as abracadabra, and all the positions in the string and their associated suffixes. And and we sort those positions based on lexicographical order of those suffixes. What we end up with might be something like this. And this has some neat properties, right? All of our A's occur together. All of our RAs occur together. Our IDs, so everything's in nice contiguous blocks, and that means we can just do a binary search to find a particular string. So in this case, if we're looking for the string bra, we can just find the midpoint. In this case, happens to be B. It's bra. So now we want to find the low point of that region. So we're going to take the thing 0, well, that's an A. So we can discard everything below that. Now we're going to take the next midpoint, ends up being 5, that's an A. Discard everything below there. So we know that A must be the bottom of the block that contains bra. So now we need to find the upper bounds. 6 is a D, so we can discard everything above. 4 is a C, and we've found the upper bounds of our block. So we've found that there's 2 occurrences of the string bra at positions 8 and 1 in the original string. And that's really good because it's a binary search. It requires m log n time. But unfortunately, it requires a lot of space. So if we're dealing with the human genome, we'd need 12 gigabytes because we're going to be using 32-bit integers to store each of those positions. Now, the paper uses something that's related to the suffix array called the Burrows-Wheeler transform, and this was invented about 6 years prior to the paper by 2 guys called Burrows and Wheeler. It's a permutation of the string, which is compressible, and it's used today in tools such as bzip, and it's also reversible. So the Burrows-Wheeler transform works like this. You take your suffixes as before, but you also append to the end of the suffix the characters that were removed. So it's basically a cyclical transform. You then sort that matrix in lexicographical order as before, and you end up with the final column, which is your Burrows-Wheeler transform. That we also add this character, the $sign, and that is a new character which is smaller than all the other characters in the string. So we end up with this funny looking permutation of the original string, and it's compressible. Right? We can use something like run length encoding, so all those A's that are in that block, we can just say there's 4 A's there, and we can say there's 2 B's. And F, which is the first column, and I like to think of it as being F for frequency, is also really compressible. Right? We can just represent the starting positions of each of those characters. So this is all really neat, but how does that relate to searching? I mean, neither of these strings look anything like the original string abracadabra. And that's the really neat thing that is described in this paper. So Ferrangino and Manzini show that it's possible to find the number of occurrences of a substring using just those 2 arrays I showed you, the transformation of the text and F of the text in OMN time. And then they show that with the help of an extra data structure, you can actually achieve OM time with some tunable space and time trade-off. And this data structure is called the FM index. FM index supposedly stands for full text index in minute space, but it's a little bit suspicious because F and M are also the initials of the author's names. So the first thing that they find is that the ranks of each of the symbols in F correspond to the ranks in BWT. So, for example, the first occurrence of the $sign in F and the first occurrence of the $sign in the transform string are the same $sign in the original text. Likewise, the first A in F and the first A in the transform correspond to the same A and S. And this is true for all the symbols. So with this knowledge, we can do what the authors describe as LF mapping. So if we wanted to reverse this, the Burrow-Wheeler transform, we could do the following. We could start at the end of the text, which we know is a $sign, and we can go to the character before it, which we know occurs in the L matrix. And that happens to be an A. So we walk to the A, and that's A1. So we now walk to A1 in F, and we know the character before that must be this R1. So now we jump to R1 in F, and we can repeat this process until we've recovered the original string. This is pretty neat. However, we had to scan through both F and L just to find the symbol with a particular rank. So we've still got this overall time complexity in the order of mn, but we can see that there's actually two fundamental operations here, rank and select. So we're going to go over doing that search again, but focusing on what we're actually achieving. So initially, what we're really saying is select from F the symbol $ that has rank 1. And then we're going to the L array, and we're saying find me the symbol at index 0. And we discover that's an A1. So then we go to the F array and select the symbol A with rank 1. And we just continue this process. So all we need to do is somehow make these rank and select operations fast. So with F, things are pretty trivial. Right, we can easily implement a function for selecting a symbol with a particular rank. All right, so if we want to select $sign with rank 1, we just look up the starting position of $, which is 0, add the rank to it, subtract 1, we get 0. We can do a similar thing for A and a similar thing for C. I think that's fairly straightforward. And it's important to note that this is complexity because we've just got a fixed set of symbols here, right? It doesn't depend on the length of the string. We can do rank in a similar fashion. L happens to be a little bit trickier. So one possibility is to have some kind of helper array, which is just a set of checkpoints. So here we've got checkpoints every 3 indices, and each of those checkpoints stores the rank up to that position for all the symbols. So position 0, A has a rank of 1, everything else is 0. At the 3rd position, A has a rank of 1, D has a rank of 1, and R have a rank of 1, etc. And so we can answer questions quite quickly. If we want to know what the rank is at index 2, well, we know it's a D. We just find the closest checkpoint, which should happen to be the one at position 3, and we can walk backwards, and we find that has a rank of 1. We could do the same for the B at the very end there. The closest checkpoint happens to be the last one. We can walk along from there. We discover it's B2. So, given we can do the rank and select now trivially for F, and we can do it for L in, with the help of a data structure using checkpoints, we've now got this ability to trade space and time to do a constant time lookup for each LF mapping step. So I mentioned earlier that it's possible to compress the string, which is quite neat. That's the opportunistic part of this data structure, by the way, the fact that if the string is compressible, the data structure can compress it. We could do something similar to what we do with the checkpoints. Here we've broken up L into groups of 3, and each of those groups of 3 can be run length encoded. In the paper they do something a little bit more complex. Rather than just run length encoding it, they use move to front encoding followed by run length encoding, and then something else. And they mention it's sufficiently simple, but it's obviously too complicated for this talk. All right, so let's revisit the backward search with these helper data structures, this time rather than actually trying to unwind the string abracadabra. Let's just find a smaller substring. So again, using bra as an example. Well, bra ends with A, so we're going to start at A, and now we know that we're looking within this region here, and we want to find the lowest ranked R and the highest ranked R in L. So we can use our helper data structure and do that very quickly. We find that R occurs at 10 and 11 in F, and now we want to find the B's with the highest and lowest rank in that region. We find them quite trivially. And so now we discover there's 2 occurrences of bra, and they occur at position 6 and 7 in F. Unfortunately, that's just the number of occurrences. It doesn't tell us where they actually occurred in the original string. That's still quite useful for a lot of problems, right? You might just want to know, does this string exist in the reference, or how unique is this string? And of course, we could do all this in some kind of compressed space, which is nice. All right, so we could reintroduce the suffix array, but that obviously takes up a lot of space. So here we can see that position 6 and 7 in F corresponded to 8 and 1 in the suffix array. But that's going to take up a lot of space. So we can use the checkpointing trick, which we just learned, with that helper data structure for determining rank and select. And by keeping every k values in the suffix array, we can again trade time and space to do lookups in the position. So here we've just discarded all the other values besides the ones that are multiples of 5. And if we wanted to find the positions for those b, we could just walk back until we end up at a symbol which has an associated suffix array position with it. So we can see here that we walk back to A's. One of those A's happen to have position 0, but the other one we're going to have to keep on walking until we hit a suffix array. All right, so we map to D, we map to A, this one here has position 5, and so from that we can determine that the positions were 1 and 8. Yep. Yeah, so we're not using any properties of the suffix array anymore. The suffix array is just there for determining the positions in the original string. The entire search is done with just the Burrow Wheels transform text and this array here, F. So we can determine how many occurrences of a substring there are in the original text with just those 2 things. And now if you want to know where they actually occurred, at what positions, then we need to have the suffix array or this reduced representation of it.
Okay. And this procedure of determining the number of occurrences is kind of analogous in the Burrow Wheels.
Yeah, exactly. So I don't know if you're familiar with the normal procedure for reversing it, where you do a set of or multiple sets of sorting. The method that Ferragina and Manzini discover is that because of the ranking of the symbols, you don't have to do all this sorting. You can just start actually at anywhere and unwind a segment. So you don't have to do an entire decompression to do a search.
And it's this property that the ranks are formulated. The corresponding symbols of the same symbol with the corresponding rank occurs like in the transform string.
Yep.
The same A, the first A in BBT corresponds to the first A in F.
That's correct, yes. Yeah,
Okay.
that's exactly it. So there's that property, and also, of course, just the fact that the character in BWT at the same index is the character which is before it. So it's just those properties that they exploit to do this LF mapping.
How does this keep the counts?
Okay, so it's a good question. Do you mind if I go back a few slides? So, all right. Right, so here we're doing the backward search. We're looking for this string bra. So we know that all the characters above A in F are characters which precede A, right? So there's some R's in there, obviously from bra. There's D's and C's, and there's the $sign. Because we're trying to find bra, we find the rank of the first R and the second R, right? And then we go into the F array, and we try and find the R's with the same rank in there, and they then point us up to B, right, the character before R that we're interested in. And we can see then that there are two occurrences of B, and they occur at positions 6 and 7 in F. Do you follow now? So because it was just like with the suffix array where it's sorted, so we're looking for blocks, right? We just need to know the start and the end of the block. The same principle applies here. We're just looking for the start and the end of the block that has bra in it.
Okay, and we have to kind of get the range. The length of this range is the number of occurrences.
Exactly. Yeah. So in this case here, the length is 2, right? The positions that those two occurrences of B1 and B2 are at 6 and 7 in the F array. And so now the issue that we're going on to is the fact that, yes, they occur at 6 and 7. There are two occurrences, but where were they in the original untransformed string? So one possibility is just to keep the entire suffix array, but as we saw, that was really expensive. You know, when you're dealing with human-sized genomes, we're talking, 10, 12 gigabytes. But we use that same trick as we did with the checkpointing for ranks, and we just take the positions at a given distance apart, so in this case 5. And now what we need to do is continue our search backwards until we find ourselves with a symbol that has a position associated to it. So we walk now to the A's that were before bra. One of those A's happened to be at 0, the beginning of abracadabra. The other one has a D before it. One had an A which has position 5, and then we can from that determine the actual position of those two strings. Right. So, I think this is pretty cool. We've got a string which is just a permutation of the original string. It's compressible, and it's possible to find the number of occurrences of a substring in OM time, right? That's pretty neat. And all we needed was this permutation and some little helper data structure for doing the rank and select queries. And of course F, which is just a few numbers, it's really small. And if we want more information, such as where those substrings actually occurred in the original string, we just need a sampled suffix array, which stores just some of the positions. Just as a quick example as to how this might perform, if we look at the human genome example, so N is 3 billion, we might have a sample checkpoints at k of 100, for example, we could then determine the positions of a particular substring in the size of the genome, which is 750 megabytes, plus 450 megabytes for the checkpointed data. Good. And if we wanted to throw in the ability to find the positions of those substrings, we could use, for example, a suffix array where we store every 100 values, and that would add an extra 120 megabytes on top. So previously we had 750 megabytes for the string and 12 gigabytes for the suffix array, and now we're down to 1.4 gigabytes. And of course you could make it smaller or larger depending on if you want to trade space for time. There's a few things I left out from the paper. Storing that suffix, that sampled suffix array, you probably noticed that they're all all those positions were just on one side of F. I think the authors use they break the positions down into blocks, and then I think they use a B-tree sort or something like that to store it. And the other thing I didn't include, which is quite important really, is a dynamic data structure. So in this example here, we built up this Burrow wheels transform text. But if you're doing that on a really big scale, it's really expensive. It takes a long time. It has to have a dynamic solution where, for example, you could add in an extra human genome or something like that. And the paper does mention an approach to that, but I haven't really looked into that. So there's a couple of reasons why the FM index might be really good for bioinformatics, and that's that biological data is really compressible. If we were to build up, say, a big database of, I don't know, criminals or something like that for forensics, humans are all pretty similar, right? I mean, we're 99% the same as monkeys, apparently, so we must be 99.99% the same as each other. So it's going to be really compressible, all that data, right? Same thing for bacteria. Lots of redundant information which can be compressed into a multi-reference database. As an example, this isn't genetic data, this is abracadabra like before, but we've also got a few mutations in abracadabra. So we've got abracadabra, adracadabra, and abracababra. And they're all concatenated, and we've spliced the $ in between. And this is how you could do a multi-string search, right? You just concatenate a bunch of strings together with $ signs in between. And here I've just done a simple run length encoding on the transformed text, and you can see it's pretty small, right? So reading this paper was really interesting, and it opens up a lot of other possibilities that I'd like to look into. So there's succinct data structures, wavelet trees, and rank select methods, and those things all kind of relate to one another. So we had that checkpoint data structure for storing the ranks and selects, but there's actually data structures where you can store a string in it, and it has the nice properties of allowing you to do rank and select on it. And you've got wavelet trees as well, which let you encode larger alphabets efficiently. So that checkpoint data structure is not going to be really efficient if you've got alphabets with hundreds of different characters in it, rather than just the 4 characters that you deal with DNA, or the 5 characters in the case of abracadabra. All right, that's it. Are there any questions? Are we all dizzy from all the LF mapping? It's OM for a sub, so where M is the length of the substring you're looking for. Plus some constants from all the extra checkpoints and mapping. Yeah.
There is one thing I understood. The checkpoint
Yes, yeah.
String.
Yeah.
But are we guaranteed when backtracking on a particular pattern to hit one of the checkpoints? Can it be that we will be hit jumping around places that don't have checkpoints? Because it's not.
So we're talking about finding the position in the original text, or so you because they're spaced. In the example, I did it every 5, right? So you're guaranteed to find it within 5 moves. Yeah, because I'm just trying to think the best way to explain it. So yeah, it's every 5 values of the yeah.
Every 5 values. So if you don't have 5 values, you have to fit one.
Exactly. Yeah. Yeah, so it's different to the checkpoints, which is based on index. This is based on value.
Okay. Yeah, that means we need some kind of data structure to efficiently find it.
Exactly. Yeah, and that's a detail I skipped. Yes. Uh-huh.
Does the sequencing technology provide free pairs? And would bow tie on this work with matching read pairs rather than single reads? We get 2 reads and then it's scanned between them.
That's a good question. I'm not entirely sure. Yeah. Yeah.
So you say it's all the end. Is that it? Is there an expectation to get faster, or the less than n?
Well, I think my understanding is it has to be n because you've got to do at least the number of symbols that are in your query string, which is n. So. Yeah, I mean, yeah, that's tunable, that constant based on checkpointing distance and that sort of thing. So with really large data sets, you're going to have checkpoints really sparse. Does that cover your question?
How much freedom is there for choosing compression algorithm? Do we need to adapt the rank select structures for every new thing that we use?
Yeah, so you can forgo the checkpointing data structure I mentioned. You could use something like the wavelet tree, which lets you encode a string as basically a series of bitmaps, and then you can use a rank select data structure to store those individual bitmaps. So that's something that people do, and if of one of those data structures, you'll get compression there. So there's a data structure called the RRR vector, which is the initials of the authors. I can't remember who they are, but that basically lets you compress bitmaps which have low entropy and do rank select queries on them.
Isn't there already some entropy compression applied to text? Right.
Yeah, so with the run length encoding, for example, yeah.
Every time you add some kind of new encoding on top, you need to adapt your rank and select procedure to work with that.
Yeah, I mean, it really depends on what the method is. So with the case of run length encoding, and the example I sort of gave here, where you break L down into blocks of 3, right? You can imagine with this one here, it's actually pretty easy to determine the ranks from those run length encoded strings, right? So once you get to one of those encoded blocks, you could easily determine what the second or first B is in that final block, for example. But yes, if you were doing something more involved, like what the authors suggest, which is move to front followed by run length, and then Yeah, I think it's some kind of variable encoding, maybe Huffman. I can't remember the details, but yes, then you'd have to do a lot more work at each of those blocks before you can determine the rank. Yeah. Yeah, that's the thing that I found really amazing with this paper is that you can take this weird permutation, which is the BWT, and then actually get back to the original string or just substrings of the original string so easily. Like it looks like a secret message, right, when you see the transform text. And it all just comes down to the fact that the ranks happen to match up in F and L. So. And also this was published 6 years after the original BWT paper, right? So it took a while for anyone to figure it out. All right. Sure. Yeah, absolutely. So bzip and bzip2 are the ones I can think of. And yeah, the compression that the authors suggest in this paper is, I think, essentially the same as the compression that's used in bzip, this three-step process of move to front, run length. All right.
Thank you.
Thank you.